welcome aboard. You know, your contribution and our contribution, we should equal them. We're all in this together. As you are aware, research on ALS is continuously progressing. Information. Wow, excuse me for a minute. We are dedicated to uncovering new discoveries, aiding in early detection, and working towards finding a cure for ALS. You can assist us, really, by participating in our speech study. The purpose of the speech study is to enhance the diagnosis and prognosis of ALS. We are seeking participants to provide 5 speech recordings, each lasting 15 minutes, once a month, over 5 months. We welcome healthy participants, as well as individuals diagnosed or probable with ALS to join us. To enroll, please visit everythingals.org/speech project and fill out the form. Thank you so much. With that, I will turn it over to our CEO and founder, Indu Navar.
Thank you, McFinn. And it's always a pleasure to host our Expert Talk series. And today we have our renowned neurologist who's internationally recognized, Angela Genge, who's joined us. And today's topic is going to be outcome measures and ALS clinical trials. A little bit about Dr. Angela Genge is internationally recognized neurologist, like I said, also well known for her work in clinical trial design and development for ALS and MND. Dr. Genge has been a director of ALS clinic at the Neuro, which is the Montreal Neurological Institute Hospital, since 1998, and executive director of ALS Global Center of Excellence in Patient Care since 2014. From 2004 to 2023, Dr. Genge was also executive director of Neuro's Clinical Research Unit. Under her direction, the CRU grew to become one of the largest neurological clinic research centers in Canada, home to first of its kind phase 1 unit dedicated to neurological disease. She's international leader in clinical trial design and development for rare neurological conditions with an emphasis on ALS and MND. She sits on a number of advisory boards as well as data and safety monitoring boards. Newfoundland-born Dr. Angela Genge completed her medical degree at the Memorial University of Newfoundland. She obtained her Canadian and American certification in internal medicine and neurology prior to completing a fellowship in neuromuscular disease at the Neuro, assisting Dr. Gordon Francis, the founder of CRU. With no further ado, I would love to invite Dr. Angela Genge to actually talk to us and just make sure that any questions you have during her presentation, as always, please put it on chat and we will do a moderated Q&A session afterwards. Dr. Genge, the floor is yours.
Thank you, Indu. Thank you, McFinn. And thank you for welcoming to this obviously very special group. I am going to share my screen and talk to you tonight about outcome measures. What I'll really try to do is go over several categories of outcome measures and really try to explain why we need to do this as part of clinical trials. The outcome measure part of any ALS clinical trial has really become much more what we call robust over the last few years, because we want to really understand the benefit of a treatment, if there is one, and make a decision about moving on from a treatment if there's no benefit. So when we look at outcome measures, what we're really looking at is ways to demonstrate that there is a positive effect of whatever drug or treatment that we're looking at. So there are many categories. I'll zoom in on a few and really talk a bit about why we do them, why they're so important, give you a couple of examples, try to do that, all of that in about 35 minutes. So, okay, now how are we going to move this on? Okay, right there. Perfect. So when it comes to outcome measures, there are a variety, but really in ALS, we zero in on a handful. We look at something called the ALS FRS, which I'm sure you all have heard of. You've probably been doing it with your nurse clinician in your clinic, even if you haven't been in a clinical trial. So I'll spend a few minutes on the ALS FRS. Survival, the importance of survival. I'll review strength testing. So, and a quick shout out to ALS quality of life measures, because in the Toferson program, in fact, the quality of life measure scales that the participants did actually signaled a benefit of the drug almost even before we saw it in some of the other measures. So it really validates the importance of that type of outcome measure in drug development in ALS. And it really speaks to how someone feels on the drug. King staging, I won't really go over. Some of these others, which are more specific, I want to mention, because if you are in a clinical trial and are contemplating a clinical trial, I want to try to emphasize why each of these things are important, even if you're having a long day or a difficult month, what you're contributing by participating in some of these assessments. So here we go. This is a pictogram, gives you an idea of how we look at it. What we are looking for is to be able to look at, let's call it the elephant, at all the different angles. So we want to look not at the clinical measurements that can tell us if there is a stabilization of a disease, an improvement in a function, evidence that on the drug you're doing better than off the drug, putting off any change in some of these outcome measures, something called time to event. We also look at biomarkers as outcome measures. If you've been following clinical trials at all, you've heard of something called neurofilament as demonstrating, even before you have a clinical benefit, a benefit to the nerves in that they're simply dying less rapidly. Target engagement outcome measures, these are ways of saying that the drug is doing what we think it's doing, and it gives us a very early signal and encourages us to move on to a bigger program. And electrophysiology. Now, anyone who has ALS has gone through something called an electromyogram or EMG. We now use components of that test, not all of it, not the needles. There are a few things we don't need to do, but components to help measure the effect the drug has in the body, even before we see the clinical impact of a drug. So all sorts of different ways of seeing the elephant. Really, every single thing we do is included in a trial to demonstrate benefit of the drug. So it's all about demonstrating benefit. So the ALS-FRSR is also called the ALS Functional Rating Scale. It's revised. It has several elements. Again, many of you may be aware of it. The questions refer to bulbar function, fine motor function, gross motor function, like going upstairs and walking, and also breathing. It's been around for a long time. It has some issues, but we're actively trying to improve how we use it in clinical trials to be able to demonstrate benefit. And we do know that the experts, the clinical trialists in the field a few years ago determined that a change of 25% in this rate of decline, so the speed with which people lose function, is clinically meaningful. Now, when I talk to patients living with ALS, some people have said a change of 1 point is clinically meaningful to them. And that is a common sentiment. So there you expect to hear a little bit more around this particular aspect of the functional rating scale. You've all done it. These are the kind of questions. This picture is just a highlight that it covers a lot of function. It doesn't necessarily cover all the things that matter to you as people living with ALS. However, it does cover functions that reflect muscle strength. And there's a real tie-in between function and strength in several of the outcome measures, because the regulators correctly see strength as something that is indicative of progression, and then maintaining strength being indicative of a response to disease or a way to slow it down. We know that the faster the change in the ALS FRSR, the decrease in survival. We know that this really slow progressors, those people who actually end up living 10, 20 years with the disease, that's also reflected in their ALS FRS. And so we're at a point now where we can start to predict how quickly someone will progress just based on their early progression rates. In contrast, and I'll be using examples from the Valor study sprinkled throughout this. In contrast, when you have a drug that works, you can see a difference in the speed of decline. And although the change in the Valor study was not extreme at 28 weeks, at every time point in the Valor study, we could see that the speed of decline of the function, so the ALS FRSR, actually was positively affected by tofersen, in that the speed of decline diminished. So the scores remained higher for longer in the ALS FRS. So the ALS FRS has issues. It has been very beneficial. We are actively working on improving it in terms of variability. And ultimately, we hope to find a replacement for that particular scale. But until we have a replacement, we will be using this functional rating scale as an outcome measure for the short to medium term in order to be able to progress drug development. But are there other outcome measures we look at? Well, strength testing is another one. It has been in vogue and out of vogue at least 3 times since I started being involved in clinical trials in ALS. Currently, it's going out of vogue. I'm sure it'll come back. The key here is that.
Can you tell me which prices are which?
Pardon? So strength testing is currently being done using techniques called handheld dynamometry. Handheld dynamometry is a device that the rater holds in their hand to assess the strength of an individual muscle. It has some issues. If your muscles are too strong, there's a ceiling effect. If the muscles are too weak, there's a floor effect. However, in the middle, it does connect with the function. So it really does reflect how much people can do. We tend to feel that it is burdensome on patients. We try now to use it only in a part of drug development where we're determining the best dose, and potentially when we're looking at a lot of different outcome measures. And we're really trying not to force people to do the strength testing in the registration trials or the last trials. So we're really working on improving this. So the reason strength is important is because in the Valor study, as we got into the 9 to 12 months, we really saw a difference. And in fact, that's when, as a doc involved in the trial, that's when I started saying, oh, this is big. And what was that I was hearing. I was having participants come back a little bit started when they were at 6 months into the trial or 6 months on drug. But once they start hitting 9, 10, 11 months, they were coming back and they were reporting functions that had previously been lost being regained in a very systematic way. And this was directly reflected in their strength testing. So for now, we still do it. We may not be doing it routinely in all trials in the near future, but you will expect to see it in certain types of trials. Another outcome measure that is now coming in the opposite direction, is becoming more common, is something called time to event. Time to event is what they use in cancer clinical trials, because the event is when the cancer shrinks, when the tumor disappears. It's really classic in developing drug in cancer. What we're starting to see is that it can do two things in our trials. One is give us a way to have to get to an answer more quickly, because a time to event the events may occur in less than a year or less than 18 months, so that we can end the trial based on the number of events as opposed to a time period. However, there's another benefit to a time to event outcome measure, and that is it can be used as a trigger to switch the few people who are on the placebo arm over to the treatment arm. So we're very conscious of how important it is to have for patients or people living with ALS to get the real drug. And we are trying to minimize the length of time that any individual could possibly be on placebo. And one of the ways that we can do this is called a rescue arm, and time to event outcomes are helping us. So you're going to start seeing that in the new trials, ways for you to, if you participate in a trial, to actually get to what we call the open label extension earlier if there's a significant, if you hit a particular event that allows us to what we call rescue that arm of the trial. Are there other things that are important? Well, I actually think this is the other big one that I personally saw in the participants in Valor that I thought was really, really important. And I've shared this anecdote a few times lately, and that's the effect on PROs. So PROs are outcome measures that are patient reported. So nothing complicated. PROs, right? It is actually how you feel as a participant in a clinical trial. And what we believe as ALS clinical trials, that PROs are actually more objective in ALS than they are in some other diseases like neuropathic pain or something where there feels to be a bigger subjective component. In ALS, it looks to be really stable and really a reflection of how you're actually doing. And to caveat this, we know that patients are people like all of us, and some people are optimists and some people are pessimists. And what we seem to see is that whatever you are in your personality, you maintain that over time. So that if you start out being a pessimist and you score yourself low, you will continue to be a pessimist. And if you improve, the score will follow the same trajectory, but you will see the improvement. And this is the same for people who are particularly generous with their scoring. What we saw with the tofersen trial is that people felt different, actually even earlier than the ALS-FRS changed and demonstrated the difference. So those progressing, the change in the rate of decline was also reflected in how people were physically responding to the treatment. And let me tell you, it wasn't because it was an easy trial. You all know that these kinds of drugs, and now there are a whole bunch more that are out there in clinical trial, are given through spinal taps. So you've got a day, every month you're coming in to get a spinal tap. It's not like giving a pill or putting on a patch. So we already know that it's a tough day, and yet still the patient-reported outcomes showed a difference, an improvement. People felt better on the drug. And a very similar program that was running simultaneously to Valor in ALS patients using lumbar punctures, we did not see the same difference. And ultimately, that treatment failed. So this seems to be pretty important. I have encouraged in one group that I am a part of to have patients, people living with ALS, really look at our PROs and see how valid they are in terms of reflecting your function, because this is something that can be improved upon for us as an outcome measure in clinical trials. Things called patient-reported outcomes. There's another category that's also super important, and sometimes people don't understand just how valuable this is. And I mentioned it earlier. It's what we call the electrophysiology. There are a number of different techniques that we use for these, and each of these techniques can be an outcome measure in a clinical trial. But electrophysiology is that EMG that you had as part of your diagnostic workup. It can also be used to follow the changes that happen to your motor system over time as you lose more motor neurons in ALS. And conversely, it can demonstrate a lack of change over time earlier than you actually see the clinical benefit. So as you hear me talk about each of these things, what we're really looking for is early signals. Signals that tell us we're on the right track. We should keep going with whatever drug or treatment we're trying. Electrophysiology fits into this. So what this EMG and nerve conduction studies do is they look at the connection, they look at the motor neuron and the connection of the motor neuron to the muscle. And these techniques are able to determine how many motor neurons approximately are still functioning. And when you compare it over time, it can demonstrate whether or not you're losing some of those neurons or whether the same numbers exist. The technique can also demonstrate if there's a lot of hyperexcitability. So if the drug calms the nerve and muscle combo down, then you can demonstrate this with this technique. And it can also show you how neurophysiology can also show how well the end of the nerve is connected to the muscle. And that should be a little bit intuitive, but a number of the new drugs are working right there, right at that connection between the end of the motor neuron and the muscle to maintain the connection. It's like putting the plug in the socket. You need the connection there in order for the action potential, the nerve signal, to actually lead to a contraction of a muscle and force. So the more nerve fibers are present, the better the connection between the nerve and the muscle, the better the strength. And so it can actually demonstrate, even before you demonstrate a stabilization in power, you can demonstrate an improvement in the electrical wiring that is our nervous system, our motor nervous system. So really important. These techniques are called CMAP, M-scan. There's another one that we use is called repetitive nerve stimulation. All of these can be used and are really good snapshots of the function of the motor nerve and the neuromuscular junction at any point in time. So there was a drug a few years ago called retigabine that was taken off the market, but now there's 2 or 3 drugs that are actually a little bit more precise and so cause less effects than retigabine that are starting to be tried for ALS. And what's important about this particular drug is it managed hyperexcitability. And so it calms the hyperexcitability, the twitching, the fasciculations, the cramps, the spasticity. All of these are forms, clinical forms of hyperexcitability. This class of drug calms that down. We were able to demonstrate in the retigabine trial that you could, by being on the drug, have a stabilization of the size of your peak CMAP. So that indicates to us that we preserved motor neurons and motor neuron function with this drug. So lots of different ways to look at what the drug is actually doing over time and giving us confidence to move that drug on to the next phase. I have to talk for a moment about neurofilaments because let's face it, it's all the rage. And it's all the rage because it was so important to the development and the approval of tofersen for SOD1 ALS. But it will be, and it's already starting to be used as an outcome measure in different phases of clinical trials to make decisions about how good the drug is. And what we're really trying to do is to put our energy into the drugs that have an effect and stop working on the drugs that have no effect. And for this, neurofilament may be helpful. If the drug that's being tried works far enough upstream, close enough to the genetic part of our system, and the products of the genetic activity, that they will directly affect neurofilament. It's not for all comers. There will be drugs that work that do not change neurofilament, but we're looking at neurofilament very specifically to help us with what we call early go, no-go decisions. So neurofilaments can help in a couple of different ways. In the really important and novel way, it can help us identify people at risk for ALS before they develop the clinical signs. Now, if you're in a family where ALS is in your family, that becomes pretty important, right? Because you may have done the genetic testing and you're carrying the gene, but you don't know if you're at risk now at 30 or you won't be at risk till 80. It'd be nice to have some signal before you'll actually get the disease so that any treatments that are approved can be used to really put off the disease in a way. Neurofilament is going to help us with that. It's being used right now with the SOD1 patients to focus in, but in this form, it's not being used as an outcome measure. It's being used almost as a diagnostic to say, okay, this person has a SOD1 genetic mutation. In this family, most people get ALS in their 50s. This person is 65 and their neurofilament is still normal. And then the next time you see them, the neurofilament levels are starting to rise. Then they're perfectly normal. But now we want to see if we can treat them to prevent the onset of ALS, the clinical signs of ALS. So big deal there. Second place it's a big deal is neurofilament can be used to separate out the different speeds of progression in a general ALS trial and sort of help us ensure that everything is balanced or choose just those patients where the disease is going to be really fast. And finally, it can be used as a traditional outcome measure, and we follow neurofilament levels to see when the neurofilament levels go down. So a lot of ways to use this. This is a blood test now. 10 years ago, it was a lumbar puncture. Now it's a blood test. Now it becomes infinitely usable in lots of different parts of clinical trials. This is just to point out what kind of things we now know. We know that neurofilament, if a high level of neurofilament means a rapid speed of progression, a low level of neurofilament, even if it's elevated, it definitely indicates a slower speed of progression. So it's rapidly become very powerful. Very, very powerful. So let's go on to something a little bit different. I'm still talking about outcome measures. The whole presentation is really outcome measures. Let's talk briefly about something called the pulmonary function test. Everyone with motor neuron disease has had a pulmonary function test because the diaphragm is a muscle, and the muscles in between your ribs that help you breathe are also muscles. So in a way, pulmonary function in ALS clinical trials is a strength test. It's a muscle function test. It is used in the clinic to determine if and when you start BiPAP or non-invasive ventilation. But it's also used for other reasons, and this is why it's important in this talk. The pulmonary function test, particularly what we call the slow vital capacity, can help us understand a few things. It can help us understand how far along someone is in their ALS, but it can also be used as a measure of strength. So there's been at least one program that showed a benefit to the loss of something called your SVC, or a lowering of your SVC, stabilized, a plateauing using their drug. So it can be used as almost a substitute, not a substitute, but in the same way that we use the ALS-FRS, and in the same way that we can use muscle strength testing, we can use the SVC to demonstrate a benefit to the drug. Because if your pulmonary function tests stabilize on the drug and and other persons don't stabilize off the drug, it can indicate really important benefit. So there have been a variety of studies using different things, but certainly they may predict survival. It may be a time to event, a milestone. It could be a time to disability. All of these can be used in a clinical trial to show efficacy or benefit from the drug. We're trying to do it remotely. We're not great at it, but we really are trying, because I don't know if you guys all realize, but we really do want to try to cut down how often you have to visit a clinic when you're in a clinical trial, just to make it easier on everybody. But what we're really trying to focus on is making it easier on the participants and their caregivers. So as we improve things like home assessments for pulmonary function, we will be able to make fewer visits, although the visits will be long, fewer visits as parts of a clinical trial. There are lots of things we're struggling with, but the biggest problem with remote pulmonary function testing is that it's not an easy thing to do. So it just is burdensome for the caregivers and the patients to do it at home without the help of a nurse or a respiratory therapist. It's not complicated. It's as simple as that. Next, and near final, is survival. Survival feels pretty abrupt as an outcome measure in ALS, even when survival also means extended use of non-invasive ventilation. The reason survival has not gone away as an outcome measure is really that it does have a clinically meaningful effect. It is very intuitive for the regulators, so people can make sense of survival as an outcome measure. In the current way that trials are done throughout the world, survival and function are both considered in looking at whether or not you have a benefit in a clinical trial. And survival is the outcome measure that the European regulators stick to very closely and really insist on in trials that are run for approval of new drugs in the European Union. So a really, really important one that we would all like to get away from. And that part of the reason we'd like to get away from it is because of the number of people you have to include in the trial. It is part of why we have to do placebo control still. We're hoping we will move away from placebo-controlled trials sooner rather than later. And certainly in early clinical trials, I am now involved in a number of programs where the placebo arm is minimal, or the period of time that the placebo controlled part of the clinical trial is very short, and then everybody goes on to the real drug. So we're working on this, but as long as we have to do placebo-controlled trials, we will be using survival as an outcome measure. So with that, I'm going to stop my presentation. I'm going to show you quickly my phenomenal clinic, and I have a whole other several other sets of slides for my other groups, but with this, I'm going to stop sharing and turning it over to McFinn and Indu. Thank you for listening.
Thank you very much, Dr. Genge. This was excellent overview of what happens during the trial. I just have to say, when I went through this with my husband, we had no idea what questions were asked and why. And this gives us an overall, a view into what happens, because as you say, we are citizen-driven research. That means we, the people, are also a very important part of research, and it's two sides of the same coin. So we all have to be educated, and thank you for this overview. Really appreciate it. And we have a lot of questions here, and so let me start with the question, and me and my colleague, Christian Rubio, will be asking the questions. And the first one is, how do we expedite the clinical trial release of the report after the trial is completed? Seems like today we have to wait for at least a year for the top line results. In especially a time-limited disease, it doesn't seem like there is an urgency, because it doesn't seem like it's fair for the ALS participants in the trial because they have to wait for so long.
So there is, thanks, Indu, and thanks for the question. So that's the question is really important, because it tells me how long it takes the information to get to the patient community. Because when a trial is actually a sponsored trial, where there is a company, large or small, that is sponsoring the trial, they actually have an obligation to release the results much sooner than a year. So let's just use tofersen. Let's use the Biogen example. They had an obligation to release that data within, I think it was about 3 months of the data lock. So as soon as they finished getting the data, they do something called a data lock. And from that point on, all sorts of people are sitting down analyzing the results, right? And they have an obligation to present that data to the public within a certain period of time. However, if a trial is run by an academic group, they do not have the same level of obligation. So, and where the results are released is actually probably the other gap. So when results are released on a sponsored trial, they are released to the investment community, because these are companies, right? They're released to the doctors who had patients in the trial, and then ultimately to the patients. But they're not necessarily released to the general public. The presentation, so a good example is the trial for the one that was called ADORE in Europe. Actually, the information was released to the investment community in the fall. It was not presented publicly at a meeting until last month. And so if it's a difficult thing. We don't do a good job of making sure that the general community knows the results of a trial. It tends to take a while, and the neurologists who present it want all this sort of information before they release the results. And maybe what we should be doing is presenting the information as we get it, and so that we learn all together at the same time. That would make a difference for the community for sure, because I've never worked with a more educated group than people living with ALS. Maybe that's something we really have to do. The absolute worst are academics. They can take forever to get the results of their trial out because they don't have the same obligation as the company. So we all have to fix them in a different way.
Yeah, it's a great point, Dr. Genge. And certainly we work with, as you know, our industry consortium is a pre-competitive group of companies who are all committed to releasing their data, sharing their data, supporting the studies we do in an open data format to advance the field and take care of people today, not 10, 15, 20 years from now. When it comes to the academics, we agree. We're certainly championing open data with them too through our Vision 2030 program. For our next question, we had a question about the ALS trial, and I believe it's an antisense oligo. So, do you know of any results announced yet for QRL 201 trial? Is it still ongoing or stopped?
Still ongoing. Results not yet. It's proceeding at a what you would consider a snail's pace, simply because it has to be done. Each person has to be done, and certain data has to be collected before the next patient is dosed. So it's really early in development. We're trying to do it as quickly as we can, but it's where we are in the development that makes it slow.
Thank you. Indy, you've got the next question?
Was asking on mute, so thank you. So the next question is more personal, where the person actually got a neurological test in the clinic that indicates that person living with ALS is stable, but he disagrees with the assessment. Is it possible that ALS-FRS doesn't reflect what a person with ALS is going through?
Oh, absolutely. There's a huge debate about that. The ALS-FRS can miss certain areas of function. It really is a very defined set of functions, right? They're the biggest set of functions, but there are a couple of people who have become people living with ALS who worked for 30, 20, 15 years in drug development, who understand the connection, and they see that there needs to be a newer scale or a more complete set of questions, so that it reflects the changes that you all are feeling that are not picked up with the ALS-FRSR. So for sure there are gaps. Maybe some of those gaps are reflected in the quality of life scores. Maybe as we develop some other measures like roads and others will be better. But I can guarantee you that you're not alone if you feel that the doctor's not seeing the changes that you're feeling when they do their scales. There's for sure that's happening.
Thank you.
Thank you. And we'll follow up with, could you share which ALS clinical trial currently excites you the most?
Oy.
You can pick more than one.
Pardon? Can I choose more than one? For different reasons. I have a top 3, top 5. I don't have a top 1. And I can honestly tell you the one thing I recommend to everybody is look at what's available to you now. Don't try to wait for 6 months to try to get into something that's coming, because you don't know what changes are going to happen that will prevent it. But Of the trials that are open or about to start, and let's put it that way, or maybe just the trials that the drugs that I'm excited about. I'm very excited. Obviously, I'm excited about the Curalis program because I wouldn't commit time to it if I wasn't really excited. I actually also feel hopeful about the drug that is currently in one of the arms of the Healy from the Calico company. I'm very hopeful and watching that program super closely. There's a third company that's working on something called PIC5, very small company. The program's not yet in the US. We're doing it in Canada and Europe. The name of the company is called Verge. Very excited to see what happens with that drug. So that's 3 right there. And then I get super excited about the really new things. So there are, now that we have really started several programs using antisense oligonucleotides, there's now the development of drugs called siRNAs. And the first ones like this were developed, the example I give best, by a company called Alnylam for a genetic disease called amyloidosis. And it's now being in the clinic. This particular type of drug is in the clinic for Alzheimer's. And it's about to go in the clinic for ALS. So top 5. I guess it's like when you choose your favorite songs, you can never come into one. There are things I'm not excited about, but these are all programs where the science is super strong and we will interrogate them really carefully. And we're getting smarter. You know, docs are like anyone. It takes a while for us to get smarter. We learn with every trial we do. So
There was a follow-up question about the Verge asset that one may not be as well known. Could you say anything further about that one?
So it targets a pathway called PIC5, and it's an oral medication. It's not an IV, and it's not a lumbar puncture. It's not delivered by lumbar puncture, but there's good rationale to think that it may actually be. There you go. So somebody's already put in the chat that it can have an important biologic difference that they'll make a change. Now there's at least another 5 that I'm also really interested in, but they're earlier in development. They're going to come up in the next year or 2. So never stop following. We don't know. I can tell you how it felt as we saw tofersen work, and it was literally in real time. If an ALS expert says to you, well, I know it wouldn't work, well, that's impossible. We don't know if it's not going to work until we do the trial. And in fact, the way that it works is you see it real time. I was involved in treating people with HIV/AIDS in the 90s. And I still to this day remember when we started triple therapy, because the men were dying and suddenly they were no longer dying. And suddenly they were living. And then they were not only living, but they were regaining function. It wasn't that somebody had a magic wand. That's why we do clinical trials, to figure it out.
Thank you.
And I think there is a just a follow-up question on what's your number 1 pick in Canada?
So we have all of these trials in Canada.
Yeah.
So you know, that little country north of the border is doing okay.
Yes.
We're very fortunate to have all of the trials and a few more running. So I really, again, it's the one that that works for you. Find the site closest to you, or just find a site that you can come to and figure it out.
Great. And the next question we got is, so additionally, what are your thoughts on the role of artificial intelligence in drug discovery for ALS?
Boy, I wish I could give you a really good answer. I can tell you that's having a huge effect. It's really, I'm definitely not an AI expert, and I can't even truly tell you how it's being used exactly, because ask Indu how it's being used. But some of the drugs that are now going to be in clinic in the next 2 years were chosen through AI technology, where I think AI in a very real sense is going to affect my work is the work that is being done to develop digital twins and surrogates, so that we can get away from, or at least dramatically diminish the number of people on placebo in an ALS clinical trial. I think that we've got to use this. This maybe even more important than some of the AI technology, but it's super important that we minimize exposure, minimize the placebo arms, so that we can get the maximum number of people on drug as early as possible in a clinical trial. And so there's a number of companies that are really helping us with this. I wish more would, because I think if we could make placebo arms the thing of the past, everyone would be happier.
Can't
Everybody,
agree more. Can't agree more. Just one of the question was on Revelio Amylyx drug. Can't even
Relivrio.
Relivrio. What have you learned from this, and any thoughts you could share?
This is a super tough one. I have a lot of thoughts. I think that the original trial showed a benefit. The second trial did not show a benefit in a larger population. That was much different than the original trial. And so what we're looking at is could we have predicted the outcome of the second trial, in which a lot of people went on it, or was there something in the first trial that led to its success? So I have a lot of thoughts, but I don't have any answers. All I can tell you is last night I went out to dinner with a colleague and friend who's also a clinical trialist in ALS, and we at our level are still talking about how to do this better the next time. So don't have great answers. I can tell you that the Amylyx company was exemplary in how they've dealt with this. Not every company would have done what they did. And they're still working on even better drugs. So we haven't lost them. They have other drugs coming along right away. But we do have to figure it out to the best of our ability to prevent this from happening again.
Thank you so much. So it's such a great answer that we're still learning, and
Yeah.
And Christian?
yeah. So Dr. Genge, are there any improvements being made to the way respiratory function is measured? Measuring devices such as the mask and the resolution don't seem as accurate with the nature of facial muscles and lung function in ALS.
Yeah, there are some improvements. It's a great question. One of the improvement may not be actually pulmonary functions. It may be that we're getting a couple of companies developing ways of measuring bulbar function by voice and other measures. And it just may be that we will be able to decrease the importance of PFTs because of the problems with seal. That's the word that we use. And so it's a really good question. I'm just very excited about modality and oral analytics and what you guys are working on, because if we can get a really good voice outcome measure, some of this lack of refinement of the way we do PFTs may become less important for us.
Thank you. And this is a very particular trial, Riluzole, a form of Riluzole that was being tested. Do you know if it's still going on?
I
Biohaven?
no, I don't. Well, I've none of the newly compounded Riluzole products have gotten to Canada. We just use Riluzole, so I don't know where that is.
Thank you.
Something similar to before, maybe. If a participant completes a clinical trial, how should one make the decision on whether they should receive the trial or continue to receive the trial, receive the trial drug or join another trial? Is there a good way to make that decision or potentially add to the regimen of a patient and family?
I think the decision becomes it's not the same in every case. For some trials, it takes a month and you can go into another trial. So if you are in a trial and you didn't really feel any change in progression, or you had a side effect and you only have to what we call wash out for a month, absolutely try a different trial. If you personally felt that there was a difference, we know that you don't know for sure because that's what placebo-controlled double-blind is all about. But if you personally felt good on the study drug, I would continue it in the open label extension because you never know, you may be in that group of people who get to be on an effective therapy 5 or 6 years before anyone else. We saw it in MS all the time, that those people who were willing to take the chance on the early trials in MS, some of them in Canada got up to 12 years of free drug.
Wow.
Because they took the chance early, felt good on it, and just stayed on the drug, and it's one of the really effective drugs. So 12 years of free drugs because they went into a 6-month or a year-long clinical trial. That adds up, right?
Thank you for that answer. And the next question is more focused on your motor neuron connection that you talked about. Is it possible that some motor neurons can reestablish connection while some of the motor neurons are still continue to die? Is this, can both of them exist at the same time?
So we don't know if it happens yet in ALS, but in theory, for sure it happens. That's what polio is. Right? Now, polio is different than ALS, and some of you may have known or had a parent or knew about polio because it was specific. They got a viral infection and the motor neurons died. But once that viral infection was turned off, the remaining motor neurons sprouted, right? Sprouted new connections, and so people regained function. If you've ever met someone with polio, if you talk to them, they may have been completely paralyzed and regained significant amounts of function. So definitely this can happen. Motor neurons have the capacity to do what we call sprout. So remaining motor neurons can expand the amount of muscle they cover so the strength comes back. The other thing that could happen as we get more effective therapies is that, and the way I describe it is you have a sick motor neuron, but if you can turn off what's making it sick, then it can regain function because it can definitely grow back the axon. We know that in a lot of ways that you can reconnect to the muscle. So it's more than not impossible. We have examples of how this works in people, in humans, in other diseases of motor neurons. So when we talk about a world without ALS or turning ALS into a chronic disease, We're talking about helping those remaining motor neurons recover to the point that even function can improve.
Thank you. And do you think, okay, and continuing the great questions, do you think we have still more to discover about neurofilaments? The phosphorylation reported is largely in the serine residue. However, no antibody is present to detect 309 and 309, is that it? And phosphorylation.
So there's a lot of work being done. We only have we only have the first layer. We know that if we can turn something off really close to the gene products, that we can decrease neurofilaments. But I can tell you there's an entire working group looking at neurofilaments and trying to understand better, either by finding newer ways of measuring that give us more precision, or really understanding why with some drug you can decrease the level of neurofilament within 12 weeks, and with another it takes a year. Because that's what's happening now. That depending on how the drug works, you may have some very different effects on neurofilament.
Thank you.
And the next question is really focused on expanded access. This person from Canada is mentioning that in US there is a better expanded access for people with ALS. Do you think Montreal or Canada will potentially develop a program similar to US's expanded access to provide access to drugs for those ineligible for clinical trials?
So the terminology is a bit of the issue. In the US, it's called expanded access, and it's evolved into this very big funded program, right? In Canada, there is another version, something called Special Access Program, that's more reflective of our healthcare system. And it is a way to get a medication outside of a clinical trial program, but is typically reserved for those drugs who have finished a clinical trial and are applying to Health Canada, for approval in Canada. A company, we can actually, the process is not company specific. What you do is you apply to Health Canada for access to a drug. Health Canada has to contact the company, and the company has to approve, and then you can get your drug. We have had expanded special access programs for Relyvrio. We've had it for Radicava. We had it years ago for Riluzole. It is normally done, though, for drugs that have just finished clinical trial, where we're waiting for the results or they're applying to Health Canada. They did it for tofersen as well. So it's not quite the same as in the US. There's no current plan that I know of from any of the companies to develop their own expanded access programs in Canada, because they have to be willing to go outside of the US.
Thank you. The next question is that the early clinical trials failure for many drugs, there have been some early or several clinical trial failures here in early in 2024. Do you think there's a loss of translation from mouse to humans, meaning, for instance, edaravone never showed survival benefits in mouse models, and based on ALS FRS, it's been approved, however, so the clinical trial of edaravone failed because it did not meet the endpoint of survival? Or, what's your take on how things are trending with endpoints and the trials that are going on this year?
So every one of these trials that have failed have had different elements. The Radicava trial in Japan demonstrated benefit on both function and survival. The study that was done in Europe was not the same as the Japanese study. So my interpretation, and I've said this publicly, is the Radicava is being prescribed for the subset of patients that was demonstrated to have benefit in the drug development in Japan, and that it's translated to the oral program because they did bioequivalency. So I've reviewed that data till the cows come home, pardon the expression. And the FDA did their own analysis, and Health Canada did their own analysis, and it is very clear that they chose a population in their study that they had evidence in previous studies were responsive, and they were able to prove. And I don't know if any of you have been on Radicava, but the people have to be early in the disease. The benefit is for those who are early in their disease. I think it's unequivocal. The study that just failed in Europe was a different drug with a different trial design and took all patients. They didn't look just at the subset that had responded to this other drug called Radicava. I don't think you can lump everything together, because what I'm seeing is we need to be smarter on drug development and really try to look at, in our registration trials, look at those patients who are most likely to respond to a drug based on your earlier trials. That's why we're doing so much work around seeing which patients respond in your phase 2 to make a really good phase 3, right? If you want to take the example of tofersen, that's what they did, but it was easy because it was genetic mutations. So they didn't try tofersen in patients with a C9orf72 mutation. They made their population homogeneous. They chose people with the mutation and had the best chance of responding to the drug. Now, it sounds very elementary the way I'm explaining it, but that really is how we're going in drug development. Find the population that the drug is most likely to respond to, and then do that in your registration trial. I don't think they had that with Amylyx. So the Amylyx drug was a bit more risky because they didn't have a subpopulation, but certainly for Radicava, there was a clear-cut population that was identified in the development in Japan. And that, it wasn't that they got a free pass. FDA looked really closely and agreed with their assessment that there was a benefit to function and survival. So I see that in all the programs I work on. We're adding in a lot more testing. So some people say, well, why do I have to do all these tests in the clinical trial? Because we need to know which people are going to respond, right? You don't want to go in a trial where you don't have a hope in hell of responding. You want to be put in a trial where you have a likely chance of responding. Am I supposed to use expressions like this on your show, Christian? I'm sorry.
100%. This is exactly why what Indu's built, and why we're here as a community is exactly for the frank conversation. And between regulatory endpoints that may not tell the whole story, and measures that don't help everything the way we want them to get us there, and then the trial designs themselves, absolutely, these are all the factors that we have to take into account, and that's what we're working on with you. So to that, I think Indu's got a good question also about those endpoints and measures.
Yeah, exactly. The next question was, could you please elaborate, which you did, but a little bit on survival endpoints versus ALS, FRS versus PRO. Can you give some examples of what a typical PRO questions would be?
So ALS, FRS is like, can you climb stairs? And I think everyone here knows what utensils can you cut your own meat, Do you have to have a caregiver cut your meat, cut your food? Survival is survival, except that also includes length of time and non-invasive ventilation as a surrogate of survival. Quality of life measures are much more about how you're feeling, how you're functioning. They're really directed at the effect on your life of the disease, the burden of the disease on your life. And I can tell you that one of the big breakthroughs for one of my patients in Tolferson was she could go up the stairs of an airplane without holding on. And she didn't realize she could do it until she did it. And it was the first thing she told me when she came to her next visit. So how that function, how that affects quality of life. I don't have the questionnaire in front of me. I'm sure that it can be distributed, but there are 2 or 3, there are even 4 different ones that are used, but it'll give you an idea of what these are like. I'm sure that Indu or Christian could get something to just show you what it's like, but I'm impressed at how sensitive they are, and they've not been considered as high on the list of outcome measures before. I think we need to move them up.
Thank you. I think we've got a couple more questions.
Okay.
Christian?
You want me to take it? Sure. So what do you think of, we've got a couple here, COIA 302 or SPG 302. What are the chances that the FDA would designate them under a fast track designation?
It might be under fast track, but they still have to be tested. So, I've never met a founder of a biotech that didn't absolutely believe in their molecule. So, and I'm okay with that because they've got to drive it. But it still has to be tested, and COIA hasn't been finished the development for sure. And the other one hasn't either. Prime C, somebody else asked about Prime C. And Prime C, we're basically waiting for funding to get to the pivotal trial, but it has some interesting outcomes in their phase 2. So things are walking through the process. COIA may be harder than some of the other trials just because of the technology required, but that doesn't mean it won't work, right? It just, the testing has to happen.
Thank you. And last question we have is, there has been a lot of press release on the first compound of NU9 that eliminates the ongoing degeneration of diseased upper motor neuron, which is a key contributor of ALS. Any thoughts on the NU9 trial?
Nothing specific, Indu. I haven't been deeply involved in that program yet, so I don't really have a good answer to that.
Thank you. Thank you very much, Dr. Genge. This was an excellent overview, and also you were so honest with all of your, answers, and so I think people here take away a lot more information than we came with. So thank you very much. And as we partner with you and other clinical trials to really make this a success and get some treatments for ALS here.
That's what we're trying to do. If we could stop the progression of the disease, that's a huge win. That's where we were with HIV. That's a huge win. And so I'm from Montreal. I saw a few people asking in the chat. So I'm at McGill. I'm from Montreal. That's where you can find me.
Thank you. Thank you very much, Dr. Genge. So we conclude that part of our evening today. And again, thank you for your time, over an hour you spent with us, and this is invaluable for us.
Everyone, this has been another extraordinary gathering. And Karen, thank you for your words of wisdom tonight. I just want to say that each one of you are more valuable than you have any idea. and you bring that value here and you share it. And so take good care of yourself so that we can meet again in the very near future and take care of each person who needs something in the square there right in front of you.
Yeah.
We care.
Right.
All right. Good night, everybody. But before we go good night, can we go say good night to whoever you'd like to say good night to? Like David Hopkins. David, good to see you. Good night to you.
Yes.
Right.
And the heart.
Patty.
Good to see you, Eric.
Good night.
Night.
Paul Fleck, Vivian.
Everybody make a heart. Where's the heart?
Okay.
Yeah.
Mark and Pam.
Oh yeah, Faith.
See you out there,
Faith
Mark.
is like love,
Thanks for
heart.
joining us. Yeah, do those hearts. Keep teaching.
Good hearts.
I'm learning.
Bye, Ted. Good night.
What a pleasure, folks.
Night,
Thank you, Keith
Gabriella.
Miller.
Bye.
And
Night, Gabriella and
Good
Brian
night,
and
Bruce.
Good night.
Night,
Do.
Kristen. And
Thanks, Faith, for coming on the show.
nice hearing.